Imagine a long, thin piece of thread. It's just a simple string. Now imagine that this string, all on its own, can fold itself into a complex three-dimensional object. A tiny, intricate machine with gears and levers in all the right places. And it has to do this in less than a second. It can't make a mistake. If it folds into the wrong shape, it's useless or even dangerous. That's basically what protein folding is. The string is a protein. Well, it's a newly made protein called a polypeptide chain. And the little beads that make up this string are called amino acids. This string can be hundreds or thousands of amino acids long. The final folded shape is the working protein. It's a hormone, an enzyme, a muscle fiber, or a part of your eye that detects light. The shape is everything. A protein's shape determines its job. If the shape is wrong, the job doesn't get done. It's like a key. If the key isn't the right shape, it won't open the lock. No exceptions. This is one of the most fundamental processes in all of biology. It's happening in your body right now, trillions of times a second. Every living thing does it. But how? How does a simple string know what shape to fold into? And how does it do it so incredibly fast? It's not magic. It's just chemistry and physics. But it's a very clever kind of chemistry and physics. Let's start with the string itself, the polypeptide chain. It's made from 20 different kinds of amino acids. Think of them as 20 different colors of beads. The instructions for which beads to use and in what order are written in your DNA. A gene is essentially a recipe for a specific sequence of amino acids. A cell reads the DNA, makes a copy called RNA, and then a machine called a ribosome reads the RNA recipe and links the amino acids together in the correct order. Outcomes are long, floppy string. This is where the magic seems to happen. The string doesn't stay a string for long. It immediately starts to fold. The secret to how it folds lies in the amino acids themselves. Each of the 20 types has a common backbone structure, which is what allows them to link together to form the chain. But each one also has a unique part that sticks out from the chain, called a side chain or R group. These side chains are what make each amino acid different. Some side chains are big and bulky. Others are small. Some carry a positive electrical charge, others carry a negative charge. And most importantly, some are attracted to water, while others are repelled by it. The ones that like water are called hydrophilic, which means water-loving. The ones that hate water are called hydrophobic or water-fearing. This distinction is the single biggest driver of protein folding. Why? Because a cell is mostly water. It's a crowded, watery environment. When the new protein string is made, it's floating in this water. The hydrophobic side chains want to get away from the water. It's like how oil and water don't mix. If you put a drop of oil in water, it forms a bead. It tries to minimize its contact with the water. The protein does the same thing. All the hydrophobic amino acids on the chain try to hide from the water. The easiest way to do that is to cluster together in the middle of the protein. They push themselves inward, creating a greasy, water-free core. At the same time, all the hydrophilic, water-loving amino acids are perfectly happy to be on the outside, interacting with the water. This process is called the hydrophobic collapse. It happens almost instantly. It's the first and most important step in folding. The long floppy chain collapses into a more compact globular structure. This isn't the final, precise shape yet. It's more of a rough draft. Scientists call it a molten globules. It has the general outline of the final protein, but the internal parts are still loose and jiggling around. So, the first big force is the hydrophobic effect. It gets the protein from a long string into a rough ball shape. But what turns that rough ball into a precise machine? This is where the other, weaker forces come into play. Once the hydrophobic core is formed, the amino acids are much closer to each other. Now they can start to form more specific interactions. One of the most important of these is the hydrogen bond. A hydrogen bond is a weak attraction between a slightly positive hydrogen atom on one molecule and a slightly negative atom, usually oxygen or nitrogen, on another. They're like tiny weak magnets. One hydrogen bond is not very strong, but a protein can form thousands of them. Altogether, they act like Velcro, holding different parts of the protein together and stabilizing its structure. These hydrogen bonds are responsible for creating regular repeating patterns within the protein. You see these patterns over and over again in different proteins. They are called secondary structures. There are two main types. The first is the alpha helix. Imagine taking a ribbon and twisting it into a spiral or a corkscrew. That's an alpha helix. The backbone of the protein chain coils around, and hydrogen bonds form between the turns of the coil, holding it in that spiral shape. It's a very stable and common structure. The second type is the beta sheet. Imagine taking a few separate strands of the protein chain and laying them side by side. Then imagine hydrogen bonds forming between these adjacent strands, linking them together like the rungs of a ladder. This creates a strong, flat sheet-like structure. It looks a bit like a pleated sheet of paper. These secondary structures, the helices and sheets, form within the molten globule. They are like the girders and support beams of a building. They give the protein its local rigidity and shape. The parts of the chain that aren't in a helix or a sheet are often called loops or turns. They are more flexible and act like hinges, connecting the more rigid secondary structures. So now we have a collapsed globule with some internal structures like helices and sheets. We are getting closer to the final shape. This overall three-dimensional arrangement of the helices, sheets, and loops is called the tertiary structure. Finalizing this tertiary structure involves a few more types of bonds. Some of the amino acid side chains are electrically charged. A positively charged side chain will be attracted to a negatively charged side chain. This forms an ionic bond or a salt bridge. It's a stronger attraction than a hydrogen bond, and it helps to lock two distant parts of the protein chain together. Then there are van der Waals forces. These are very weak, very short-range attractions that occur between any two atoms that are close together. They are a result of fluctuating electrical charges. Individually, they are almost insignificant. But inside a tightly packed protein core, there are countless van der Waals interactions. They are the final bit of stickiness that helps all the atoms nestle into the most comfortable low-energy positions. Finally, there's a special kind of bond called a disulfide bond. This one is different because it's a covalent bond, which is a much stronger, more permanent chemical bond. It can only form between two specific amino acids called cysteine. If two cysteines from different parts of the protein chain come close to each other, their side chains can link together, forming a strong staple that locks the protein shape in place. Not all proteins have disulfide bonds, but for those that do, they provide a lot of extra stability. This is especially important for proteins that have to exist outside of the cell in a harsher environment. So, to recap the process, the chain is made. It immediately collapses due to the hydrophobic effect, forming a molten globule. Inside this globule, secondary structures like alpha helices and beta sheets form, stabilized by hydrogen bonds. Then, the whole thing settles into its final, precise tertiary structure, locked in place by a combination of ionic bonds, van der Waals forces, and sometimes disulfide bonds. The final shape is the one with the lowest possible energy. Think of it like a ball rolling down a hill into a valley. The unfolded chain is at the top of the hill with high energy. It can roll down in many different ways, but it will always end up at the very bottom of the valley, in the most stable, lowest energy state. This energy landscape is often visualized as a funnel. The wide top of the funnel represents all the possible unfolded shapes. As the protein folds, it goes down the funnel, having fewer and fewer possible shapes, until it reaches the single native structure at the bottom. This funnel idea helps explain one of the biggest paradoxes in this field. It's called Levinthal's paradox. A scientist named Cyrus Levinthal calculated that if a protein tried to find its correct shape by randomly trying every single possible conformation, it would take longer than the age of the universe. But we know proteins fold in microseconds or seconds. They clearly don't do a random search. The energy funnel shows how they are guided. The hydrophobic collapse rapidly narrows down the possibilities, and then the local interactions guide it quickly down the funnel to the bottom. It's not a search, it's a guided collapse. Some very large or complex proteins have an extra level of structure. This is called quaternary structure. It's when multiple already folded polypeptide chains, each with its own tertiary structure, come together to form a larger protein complex. Hemoglobin, the protein in your blood that carries oxygen, is a classic example. It's made of 4 separate protein chains that fit together perfectly to do their job. The forces holding these chains together are the same ones we've already discussed. Now, this process sounds very neat and tidy, but what happens when it goes wrong? The cell is a very crowded place. A newly made protein chain is sticky. Its exposed hydrophobic parts could easily glom onto another sticky, unfolded protein. If this happens, they can form a useless, tangled mess called an aggregate. This is a big problem for the cell. To prevent this, cells have a system of helper proteins called molecular chaperones. You can think of them as the quality control managers of the protein folding factory. One major type of chaperone works like a little isolation chamber. A well-known example is the GREL-GROS system in bacteria. When an unfolded or misfolded protein comes along, the GRO-EL chaperone, which is shaped like a hollow barrel, captures it. The protein goes inside the barrel, and a lid GRO-S closes on top. Inside this chamber, the protein is safe from other proteins. It has a private space to try and fold correctly. The walls of the chamber can also help nudge it in the right direction. After a few seconds, the lid comes off, and the now correctly folded protein is released. If it's still not folded right, it might get captured again for another try. Other chaperones, often called heat shock proteins because their numbers increase when a cell is stressed by heat, work differently. They can bind directly to the sticky hydrophobic patches on an unfolded protein, preventing it from aggregating with others. They act like a shield until the protein has had a chance to fold properly. They can also help unfold a protein that has misfolded, giving it a second chance to get it right. But even with chaperones, mistakes can happen, and when protein folding goes seriously wrong, it can lead to devastating diseases. Many neurodegenerative diseases, like Alzheimer's, Parkinson's, and Huntington's disease, are now understood to be protein misfolding diseases. In Alzheimer's disease, a protein called amyloid beta is the culprit. Normally, it's a small, soluble protein that gets cleared away. But in people with the disease, this protein misfolds. Instead of staying soluble, the misfolded proteins start sticking to each other. They form long, insoluble fibers called amyloid fibrils. These fibrils clump together to form the infamous plaques that are found in the brains of Alzheimer's patients. These plaques are toxic to brain cells and disrupt communication between neurons, leading to memory loss and cognitive decline. A similar thing happens in Parkinson's disease, but with a different protein called alpha-synuclein. And in Huntington's disease, it's a protein called huntingtin. In each case, a specific protein misfolds and aggregates, forming toxic clumps that damage cells. There's also a category of diseases called prion diseases, like Creutzfeldt-Jakob disease in humans and mad cow disease in cattle. These are particularly scary. The culprit is a prion protein. We all have normal prion proteins in our brains. But this protein can misfold into a toxic, infectious shape. The truly terrifying part is that this misfolded prion can act as a template. When it bumps into a normal prion protein, it can cause the normal protein to flip into the wrong shape as well. This sets off a chain reaction, where more and more proteins are converted to the misfolded, aggregate-prone form, leading to widespread brain damage. This shows just how critical correct protein folding is. A single protein, with the exact same sequence of amino acids, can be either a vital cellular worker or a deadly poison, all depending on its three-dimensional shape. For decades, one of the biggest goals in biology has been to predict a protein's 3D structure just from its amino acid sequence. This is known as the protein folding problem. If we could do this accurately, it would be revolutionary. We could understand what thousands of unknown proteins do. We could design new drugs that fit perfectly into the shape of a disease-causing protein to block it. We could even design brand new proteins from scratch to act as medicines or industrial catalysts. For a long time, progress was slow. Scientists used techniques like X-ray crystallography and cryo-electron microscopy to figure out protein structures experimentally. These methods are powerful, but they are also slow, expensive, and don't work for every protein. Predicting the structure computationally was a huge challenge, but recently there has been a massive breakthrough. Artificial intelligence programs, most notably DeepMind's AlphaFold, have changed the game. These systems have been trained on the vast database of known protein structures. They use deep learning to recognize the complex patterns in an amino acid sequence that determine how it will fold. AlphaFold can now predict the structure of most proteins with incredible accuracy, often matching the results from years of painstaking lab work. It's like having a cheat code for biology. Scientists can now get a highly accurate 3D model of a protein in minutes just by feeding its amino acid sequence into the program. This is accelerating research in every area of biology, from understanding diseases to developing new technologies. This doesn't mean the protein folding problem is completely solved. We still have a lot to learn about the dynamics of folding, how proteins interact with each other, and how to predict the structures of large, complex protein machines. But we've taken a giant leap forward. So when we look at how a protein folds, we're seeing a beautiful display of physics and chemistry at work. It starts with a simple genetic code that specifies a one-dimensional string of amino acids. This string, guided by the fundamental desire of its hydrophobic parts to escape from water, collapses. Then, a delicate dance of tiny forces, hydrogen bonds, ionic attractions, and more guides it to fold into a single, specific, functional shape. It's a process that is both robust and fragile. It happens with incredible reliability, trillions of times a day, building the machinery of life. But when it fails, the consequences can be severe, leading to some of our most feared diseases. Understanding this process is not just an academic puzzle, it's a direct window into the workings of life itself, and it holds the key to understanding and potentially curing a vast range of human illnesses. The simple string becomes the complex machine, and in that transformation we find the very essence of what it means to be alive.